other, and that gives rise to differences in translation of whole classes of proteins that have that particular codon in them. And these would be kind of invisible to an expression array study, because many of these SNPs would actually not exist in the mRNA that for the target you're studying. They exist in something that regulates the translation of all proteins, right? So just to briefly give kind of a rundown of kind of the methods that are used to do this, or have been used to do this in the past, and why it's been difficult to scale these up to whole genome studies. There's the traditional 2D gels and mass spec. This is kind of what I got a little bit of training as a biochemistry graduate student a long time ago, and I thought, wow, this is really a tough job to cut out lots of bands and put them in the mass spectrometer and try and recast the gels and get the same experiment several times. It's really a tough chore if you were to try and do a protein phenotype on 100 individuals and try and understand the genetic determinants behind these spots. It's very difficult to quantify some of these, and it's very difficult to go across many samples. So there's newer methods that have been developed in the last 10 years, one of which is the so-called reverse phase lysate array that's been pioneered by Lance Leota and Emmanuel Petricoin, and now has also been pushed forward by Gordon Mills at MD Anderson Cancer Center. The notion is you can take cell lysates from your cells of interest that have been treated with different conditions and array those cells onto so-called reverse phase lysate arrays. These are just nitrocellulose coated slides that you can array your lysates on, and then you add antibodies to proteins of interest. And the notion is if your protein of interest goes up or down or gets modified in a certain way, and if you have an antibody directed at that protein or modification, you can quantify it and you can use very small amounts of sample. But the real problem here is that there's a lot of cross-reactivity that goes on with antibodies, and even though you've purchased an antibody to protein 1, it may recognize protein 1 really well, but it may also recognize a little bit proteins 2 through 50 that would obscure your ability to see changes in protein 1 because there's background there. So there are a limited number of antibodies that you can use with the approach that are highly specific, highly selective, and yet there's a lot of validation that goes into that process of finding the antibodies to begin with. So the kind of tried and true method that's been used for a long period of time is the immunoblotting or Western blotting method, where you electrophorese samples on a gel to reduce the complexity of the mixture. And then even if your antibody kind of has some other bands that show up, you can kind of, in many cases, discriminate between what you know is the band you want to look at and the other bands. But it's a lot of work to measure each individual protein. It takes a lot of reagents. It's expensive. Each one of these antibodies now is on the order of $300 to $400 for 100 microliters, and that's enough to do 5 to 10 experiments. If you wanted to go across all transcription factors across hundreds of individuals, this would be impractical. So as a result, most proteomic studies trying to relate genetic differences in your predisposition to disease or cancer have either examined small numbers of proteins across, let's say, the NCI 60 panel, or across lung cancer samples, etc. Or they've examined a large number of proteins using mass spectrometry across 1 human cell line or across 3 human cell lines and tried to ask, are there differential expression? And with these 1 or 3, you can never have any power to say this genetic variant is responsible for this difference in your phenotype. And if you're going across samples, just small numbers of proteins, it's really difficult, in fact, to discover new variants associated with response, let's say, to chemotherapeutic agent. So the few studies that have kind of pushed this forward, trying to understand relationships of genetic variation differentially with mRNA and with proteins, they've used yeast as a model system. And in those particular cases, they found loci that correspond independently to protein expression. They found loci that associate with transcripts. And they've also found loci that affect both, but differentially. That in many cases, the effect on one is much greater or different than the other. And this information suggested to us that if we were able to measure large numbers of proteins across large numbers of individuals, we might find unique loci that would give rise to differences in proteins. And in fact, those loci may also have been previously proposed to be important for your response to diseases. And therefore, you might infer that those proteins associated with the loci that were responsible with the diseases might be drug targets for that disease. They may actually causally affect that disease. And if you could find 10 of those proteins associated with different loci associated with disease, you might be able to start building networks of influence of proteins related to disease. So we thought, so I'm in the Binmay department and a close collaborator physically, and now in terms of our work together is Eileen Dolan. She's done a lot of work on relating genetic variability with your differential ability to respond to common chemotherapeutic agents that are used to treat cancer, like cisplatin, carboplatin, 4-HC, etc. And she's done a lot of work to relate genetic variants that associate with that, and so we thought we could actually start to relate protein levels to genome loci. There are many genome loci that have been associated with response to chemotherapeutic agents, but in many cases, the false discovery rate associated with those associations is very high. Maybe that 90% of the variants you found are false positives. And the reason is because there are so many loci, in many cases 10 million SNPs, that you're testing for association that simply by chance you'll pick up many false positives. But the notion is if you have a series of SNPs associated with protein expression, and you have a series of proteins associated that we found whose levels are associated with chemotherapeutic agents, some of which you knock down and you show that now you've induced changes in sensitivity to agents, you might actually be able to narrow down the search of genome loci associated with responses, and you may be able to find new proteins associated with responses. So here's the cast. Ron Hawes is a crazy graduate student that came to my lab and said, look, I'm trained in plant genetics, and I'd like to actually start doing genome-wide protein studies, protein associations. And I have a little biochemistry experience, but if you could teach me biochemistry, I have a lot of genetics experience, and so we could kind of start bridging the gap. Amy Stark is a graduate student in Eileen Dolan's lab. She actually just got her Ph.D., and she's very interested in the genetic determinants of pharmacologic response. Kevin White, I'm not sure if I should call him crazy or not. He's the director of our institute, and he was responsible in large part for recruiting me, and the notion he gave to me was, Rich, if you could find some way of measuring all the human transcription factors. We have these RNA sequencing methods and nanostring and other things where we can follow mRNAs, but we don't really know what proteins are responsible for transcribing those mRNAs. We're inferring, and we're trying to do computational ways of doing that, but if you could have some method of doing that, we could potentially start to really increase our understanding of transcriptional responses. So with Kevin's help, we purchased an initial collection of about 5,000 antibodies directed at all human, in fact, multiple antibodies directed at all human transcription factors. And the notion was we would start to use our methods to screen those antibodies for which were the good ones, which were the bad ones, and then we would use that collection to start screening HapMap cell lines for expression of large numbers of transcription factors and relate them to these chemotherapeutic differences. And so I'll back up a little bit. For those of you that aren't aware of the HapMap project, the notion is over the last 10 years or so, blood samples have been collected from individual populations across the earth. There are Asians and Africans and Chinese and Japanese and Ceph population in Utah. The notion is that these B cells were taken and then immortalized with Epstein-Barr virus and given out to labs all across the world where their genome was profiled for large numbers of SNPs. And I'll talk a bit more about that. And there are advantages and disadvantages to this particular system, and we think many of the advantages outweigh the disadvantages, and I'll talk about many of the advantages. It's very easy to experimentally manipulate them. You can very quickly take these panels of individual cells and actually profile their response to different agents. And now they've been genotyped, and they're fairly cheap to grow. They're unlimited resource because they've been transformed with EBV. And there's lots of genetic information, and I'll talk about more information that's available for those in just a bit. On the downside, the phenotype may not recapitulate in vivo. This is an in vitro system subject to in vitro confounders. Many of the cells have different numbers of Epstein-Barr virus that have integrated into them, and some of them grow slightly faster than others, and so in many cases we have to perform surrogate variable analysis or principal component analysis to try and regress out non-genetic confounders. Most cell lines lack P450 system for pharmacokinetic studies, and so that's another limitation. So here are the things that are available with this particular system that made it pretty attractive for us. There are 4 million SNPs directly genotyped, and with new 1,000 genomes data, you can impute much of the missing data, which allows you to have about 12 million SNPs at a minor allele frequency of greater than 0.05. There are expression arrays, Affymetrix exon arrays that have profiled the expression of these individuals in culture. There's RNA sequencing now from Jonathan Pritchard's lab at University of Chicago. Our collaborator, Eileen Dolan, has profiled now the microRNA in addition to mRNA with a postdoc in her lab, Stephanie Wong. Eileen has now measured the response of these cells to 18 different drugs. And so we can actually ask what proteins are associated with cisplatin, sensitivity, carboplatin, et cetera, for 18 different drug treatments. We have a lot of knowledge about non-genetic confounders from other laboratories that have used these cells. And now we also have a lot of SNPs that are associated with many complex diseases. So if we find a SNP or a series of SNPs associated with a series of novel transcription factors, and those same SNPs are associated with complex disease traits, we can potentially make the gap or bridge the gap and say maybe these proteins associated with those loci are also associated with those disease phenotypes. It's hypothesis generating from that perspective. So to stop for a second and ask a few overarching questions. How can we measure thousands of protein levels across human populations, or a human population of 68 lymphoblastoid cell lines from the Yoruban population of HapMap? Is the genetic variation associated with mRNA expression the same? Are there the same SNPs that if you regulate an mRNA abundance in cells in culture, are they also regulating the proteins, or are there different SNPs for each? Which proteins are associated with resistance to chemotherapeutic agents? This is really cool. There's a lot about what's known about, let's say, cisplatin damaging DNA, and how you can detoxify cisplatin, or how you can detoxify cytarabine through, the cell has mechanisms for converting these particular agents into other agents that are less toxic. There's a lot regarding cell signaling, and if a cell should choose to die, or if it should choose not to die, go into a quiescent state. There are many things that aren't exactly known about how that works. Can we find genetic variants previously associated with complex traits that are associated with protein expression? There are lots of these. There are lots of SNPs associated with particular phenotypes for which they're in gene deserts. Maybe they're in a non-coding RNA. We don't know. But perhaps we can actually start to relate some of these to protein levels and actually find out what the underlying protein mechanisms are. So the first question, how can we measure thousands of proteins across human populations of 68 LCLs? So here I'll take a step back from all this talk about genetics, and I'll talk about the basic laboratory problem of how do you measure proteins and measure it in a quantitative way, in a way that has a large linear dynamic range and doesn't require thousands of graduate student and postdoc hours and all the agony that goes along with measuring proteins. So my advisor, Gavin Macbeth at Harvard University, as a postdoc, when I was leaving, he had published a paper suggesting that if you take cells, you grow them in culture, you lyse them, and you array them onto those slides like I mentioned a few minutes ago, called the reverse phase lysate array. And it's kind of an unfortunate word. It's reverse phase because originally people put antibodies on slides and pulled them down with arrays. And now with the new method that came out in the year 2000, they reversed that, and they're just putting lysates directly onto pieces of nitrocellulose, and so they call that reverse. Notion is now you can add antibody to protein 1, antibody to protein 2. These can be pan antibodies, meaning that they don't care about modification state. Or you can buy antibodies that really do care about specific modification states, and you can profile those across large numbers of slides using a minimal antibody and using minimal cell reagents. And he wanted to use this to profile pathways, but there were some big problems along the way. If you look at this Western blot to alpha phosphothreonine 308 AKT prior to or after stimulation with EGF, you can see at time point 1 you see great phosphorylation increase on threonine 308, but you have this background thing that doesn't change. It's just giving you signal regardless of the change in the other signal. So if you go down here and look at this graph, this is the signal before stimulation on the array, meaning that this band here actually gave a lot of signal without regard to the phosphorylation state of this protein, versus the Western blot. The Western blot showed a huge change on the blue. This array didn't because of this background problem. And he went through lots and lots and lots of antibodies. He published 34. He actually, I think, originally tried about 340. The ones he published were these. 4 of 34 antibodies generated equivalent identical information, meaning that antibody was so specific that it was giving you very quantitative information on the one thing you were looking for. 12 of 34 antibodies generated similar trends, so you kind of could see something changing, but now the dynamic range was so compressed that it was much less than before. This is a huge problem, and it raised a lot of alarms, and many of the people in performing the ART kind of knew about this. You have to screen 1,000 antibodies to get your good 20 or 30, and that's just something that they did. You just screen a lot of antibodies to find a small number, and I thought this is actually a problem that's basically prohibiting the field. Nobody can move forward if you're stuck on 20 antibodies, some of which are related to that thing you want to study, and most of which are not. So I had the notion if you could just somehow run a Western blot, it didn't have to have much resolution, but if you could get a little bit of protein separation, maybe you could get over this problem of cross-reactivity. So our notion was you don't print directly onto nitrocellulose, you print onto acrylamide gels. And now you actually put a buffer strip here. So this is a non-contact microarray. It's picking up like a microliter of your cell lysate, and then it's depositing a few hundred picoliters across the gel. So in this case we have 6 samples that have been treated with EGF. We've lysed them. We're printing a protein ladder and those 6 samples, and we put a buffer strip at the top and the bottom. These proteins are just sitting on the top of the gel. They kind of absorb a little bit into the gel, and after you put the buffer strip on and apply a voltage, they'll all go in this direction downward. And so you can see here, after you've electrophoresed with a semi-dry electrophoresis, you can see the protein standard and the bromophenol blue at the bottom. And this is the equivalent of 96 individual little Western blots. And so after you've electrophoresed them, you transfer them to a piece of nitrocellulose, put this under a gasket and scan the membrane. Well, first you need to add an antibody that has a fluorescent label, or a secondary that has a fluorescent label. And then this is what that looks like. These are a whole bunch of beta-actins. This is all 6 time points for replicate 1 and 2 and 3 and 4 and 5, the same cell lysates, same 6 cell lysates from different time courses, all blotted with beta-actin to show the same loading. And then this is a whole bunch, 96 different phosphoantibodies that are detecting different things. In this case, you see phosphoepidermal growth factor receptor tyrosine 1068 that gets phosphorylated immediately within a minute after adding epidermal growth factor. But then you see other proteins that respond with completely different kinetics because they're performing different functions in the cell. For example, the S6 ribosomal protein responds very slowly after you change the growth media and you have new amino acids, new glucose. You'll probably activate mTOR and AKT, which starts to ramp up the metabolic cycle of the cell, and you'll start to see the ribosome get phosphorylated, indicating increases in nutrition. So we did a lot of proof of concept in this first Nature Methods paper to show, look, you get really good resolutions, similar to a standard new page gel from Invitrogen. You can easily resolve 25, 37, 50, depending on how long you run out these microwesterns. And if you simply load a protein ladder, and do twofold serial dilution, you can see linear response of many different proteins. If you do the same thing with a whole bunch of cell lysates and dilute them twofold and then add many different antibodies, you can see a linear response of all the antibodies, and you can pick out the particular proteins of interest you're looking at, without regard to the other background protein spots. And we did a whole bunch of comparisons side by side of a standard Western blot with a micro-Western blot and compared time courses. And in all the cases we studied, the time courses were very similar across them. In fact, we picked 2 from the Macbeth paper that showed that the antibodies were good, and they were actually good in our case too. And we also picked many of the ones that actually did not respond with the reverse phase array. And they actually work okay with reverse phase lysate array, because in many cases you can see background bands and you simply can ignore them. You don't have to quantify them if you don't want to. And this enabled us to generate a whole lot of phospho signaling data. This is, to my knowledge, the largest number of phospho proteins that have ever been profiled or examined with antibody-based methods. And so we stimulated cells with different levels of EGF, and then we followed all the phosphorylation patterns in time, and we classified lots of membrane proteins that are other receptor tyrosine kinases that were getting phosphorylated very early. And then we had mid-stage of MAP kinases and ribosome response regulators. And then finally, at the very end, you see a lot more ribosome proteins and transcription factors being phosphorylated. So the notion is we could use this method to profile many proteins, unprecedented number of proteins with antibodies pulled off the shelf from catalogs from vendors. And you only need to use a few nanoliters for each measurement, so you don't need to worry so much in a discovery-oriented screen for asking, what's the target of my small molecule? I'll just add the small molecule, and I'll see, aha, these proteins, they all went down or they went up after some time with my molecule versus without my molecule. So we worked with Doug Laufenberger at MIT to start to connect the dots. What are the proteins that are causally responsible for phosphorylating each other? And I don't have time to go into this, but the notion was this was kind of an initial phase of how would you collect a lot of protein data. If you had a lot of protein data, how could you make sense of it and turn a whole bunch of heat map numbers into this is upstream of that and it's upstream of that. I predict if I added an inhibitor to this, then this should go down, but then there's this feedback mechanism that may change, right? The notion is we want to start putting these together and making sense of the data. So about towards the end of the development of this method is when Ron Haas joined the lab and suggested that, and was talking to others in the human genetics department that suggested this would be really powerful if you could start to profile across the genome large numbers of transcription factors and cell signaling proteins. So we thought we would use this method, micro-Western arrays, to quantify the steady state abundance of several thousand proteins in the Reuben population of HapMap. We would initially focus on transcription factors and cell signaling proteins. We would then relate these abundances to genome sequence, other central dogma intermediates like mRNA, microRNA, as well as a phenotype, the sensitivity of these cells to killing by chemotherapeutic agents. So we're starting measuring proteins, going to start comparing them to genome variants that are, and see if the same ones affect mRNA versus protein, and ask which of the proteins associated with chemotherapeutic sensitivity. So we have a pipeline. We start out with 3 thaws from each of 68 unrelated Reuben individual or Reuben LCLs. So the notion is technicians pull 68 HapMap cell lines. These are from 68 different people. Pull them out of the freezer. They grow up big bottles of them. They then freeze pellet those 68 samples, make 30 aliquots of each of them. For our study and for each of our experiments, we pull out one of the aliquots to perform our proteome-wide analysis of transcription factors. In the initial stage, we had to find out first which of all the antibodies we have actually work. So we pulled the sample and we began screening these initial 4,366 antibodies, and we asked, did we see a band? Did we see what looked like a band at the initial starting concentration of this antibody? Well, if it gave something like this, we would go back and repeat the experiment, but we would use 10 or 100-fold less of the antibody. And we're simply asking, which of these antibodies give any signal if in pools of 10 individual cell lines? And so then we start checking off the box. This antibody seems to work. It gave a signal. We don't know if that signal really is recognizing the protein we think it is, but in the end, if it's a really interesting correlation that we find about that protein, we'll knock it down and ask, did the band go away? And that will tell us this antibody really was recognizing the protein we think it is. So if the antibody gave a single band, we could only see one band that was very high signal, we would then go back and take the whole 68 individuals and we would quantify them with this method that I told you that has problems with antibodies that don't generate just generate single bands. If there were multiple bands, then we would take all 68 back through the micro-Western array method and quantify all 68 individuals with this method. And then at the end of the day, we ended up with a matrix out of the original 4,366 with a matrix of 465 protein expression levels per individual across 3 biological replicates. And then this whole micro-Western array experiment was also repeated 3 times, so we have an idea of the technical variation as well as biological variation. And then the next stage of this was to further validate the antibodies we think are the good ones by knocking down a subset of them and then asking the question, which of the bands, what percentage of the bands actually went away. So the notion is now at the end of the day, we have all of these data sets that have already been generated by others on HapMap. We have now our expression of 365 transcription factors. Based on RNA sequencing, there should have been 1,000, we think, expressed in LCLs it's always difficult, but if you pick a threshold of like 10 reads per million RPKM, reads per 1,000, in any event, there should have been 1,000 mRNAs expressed by RNA sequencing. We found 365 transcription factors, and we found 100 cell signaling proteins that were expressed. And many of these are the total abundances. In many cases, it's actually the modification states of signaling proteins. So I know my time is running out, so I will, this is all fairly new and we're just writing it up now for publication. But we identified 579 loci associated in cis with 81 protein levels. That means that there was a single nucleotide polymorphism that was located in the gene model for 81 of the proteins whose expression seemed to be highly associated with those. And so here's just at the top of the list as far as alphabetically. There are SNPs associated with the expression of ribosomal components, transcription factors, etc. Lots of different proteins. And we found that up to 50% of all cis eQTLs, that means someone in the past has found a loci that was associated with the mRNA expression of a gene. And we found that 50% of all of the SNPs that were associated with mRNA expression were also associated with the downstream protein expression. And depending on the p-value you pick, once you get to the highest p-values of RNA sequencing, those are the ones where we start to really pick up, which means potentially many of these that have previously been found by mRNA expression at lower p-values, many of them are actually false positives. And we identified over 100,000 so-called trans-pQTLs. These are things located throughout the genome that are nowhere close to the protein that they're regulating. But nonetheless, at 4 times 10 to the minus 11th, this is genome-wide significant. Most of these actually at the top are genome-wide significant. And these, to my knowledge, are the first ever trans loci in the genome that have ever been associated with protein expression. So the notion is, for example, if you have a certain allele at this position of chromosome 16, you'll have greatly different levels of the Dido-1 protein, and it's on a completely different chromosome. So here's an example of a replicating eQTL pQTL. So with this particular loci and ZNF266 at the mRNA level by RNA-seq and expression arrays, you see the CC individuals have lower levels of the mRNA, and if you use Western blots or these micro-Western blots, you see differences in the protein expression in the same direction. And so we also identified many novel protein-specific QTLs. If you, for some technical reason, there were very few reads actually picked up at the mRNA level on RNA sequencing, and now we think, in fact, many of these early RNA sequencing studies didn't sequence to enough depth. So in fact, there are many low abundance mRNAs that you actually cannot read by RNA-seq that actually give very high signal at the protein level. And in some cases, the SNP is actually in some of the probes on the expression arrays as well that were thrown out because of potential mismatches. So here's an example of a trans SNP. If you go across, these are recombination blocks in the genome, and then we found there are a series of SNPs in this particular recombination block that are highly associated with DIDO1. DIDO1 is located on a completely different chromosome. And this SNP is located in between two genes, one of which is a lysyl tRNA synthetase and one of which is an adenosine deaminase, both of which are responsible for the charging of lysine-specific tRNAs. So the notion is, if you regulate the expression of a tRNA, or the expression of, or the function of a tRNA synthetase, perhaps you may also regulate the expression of many proteins downstream that have that particular lysine codon in them, and that would be invisible to you by an mRNA expression array analysis, or expression analysis. So we've identified hundreds of pQTLs that overlap with previously described SNPs associated with disease traits. Many of these are kind of interesting and would be expected. There's a SNP that we found associated with GAB1 levels, and that's GAB1 is the adaptive protein for hepatocyte growth factor. This SNP has been shown to cause you to have a different disposition to hepatocellular carcinoma. HOXB7, which is important for vertebrate patterning, is also associated with a SNP that has been previously associated with hip geometry. And so we have a series of these, whole bunches of which for lots of different disease traits that we're now following up on to ask the question, is this protein TUB that is associated with the SNP associated with adiposity, and this TUB protein has been associated, if you knock it out in mice, it affects their fat metabolism. So potentially now we've actually identified the underlying variant, the underlying reason why potential SNPs give rise to differences in phenotype. And then lastly, and this is I think really cool, we've identified about 200 proteins associated with sensitivity of cells to 18 chemotherapeutic agents. So depending on, so I told you that our collaborator Eileen had profiled the chemotherapeutic sensitivity of 18 different agents, and with each of these agents we've identified between 18 and 30 novel proteins that are highly correlated with those responses. So here are just a few of them, and the notion is now, if we're going back again and saying, if we have a protein associated with a particular response, and that protein is associated with a SNP, is a SNP associated with the response, and if we get this full triangular pattern relationship, it gives us high confidence that we've identified a bona fide genetic variant associated with your response to a particular therapy, and we've identified the protein that is mediating that response so that particularly you could actually make a drug to modify that response. So here's an example, and I'll end very soon after this. So the notion is we found SNP associated with Ara-C sensitivity at a certain p-value. We also found this SNP was associated with ATF2, a DNA damage response protein. And we found this DNA damage response protein associated with the sensitivity of cells to Ara-C. So we thought, okay, well, if you knock down ATF2, you should make the cells more sensitive to apoptosis. So we first asked, can we actually measure ATF2 being knocked down by siRNA? And we found 25 hours that worked the best. So we picked 25 hours as the time we would test for differential sensitivity to Ara-C. And so we have two different siRNAs here. The red is the siRNA to ATF2, and the black is the scrambled. And in both cases, you see a subtle effect, although it's statistically significant, that shows that, yes, indeed, ATF2, if you knock it down, you will get greater sensitivity to the chemotherapeutic agent. Similarly, with paclitaxel, we found that for DIDO1, it's highly associated with apoptosis. If you have higher levels of it, you'll have higher apoptosis, and if you knock it down, you'll get lower apoptosis. This was the so-called functional validation of the associations that we're finding. So finally, we found also a whole lot of microRNAs that are associated with protein expression. This HSA MIR491-3P, highly associated with the protein expression levels and has no association with mRNA expression levels. So here's ZNF7, one of the associated proteins, has a high association with this microRNA expression, and it has no association with the mRNA expression, suggesting that this is potentially regulating the translation of this particular mRNA. So I'm not going to go through all the potential future directions. You can imagine if you can study lots of proteins, you can start to relate it to whole lots of different things that I think are pretty new and exciting. And we're really excited about this. Going to chip lots of transcription factors and find where they're binding in the human genome, relate those to human disease. This is just an example of recent use of the method that came out in Cell a few months ago by Aviv Regev and Nir Hakohen at Harvard Medical School basically to ask how the signaling pathways are affecting the maturation of dendritic cells. And with that, I'll end and thank Mark Chacho and Max Chu, the co-inventors of the method, and Amy Stark and Ron Haas, the graduate students that were responsible for doing the pharmacology and protein analysis work. Kevin White, who is the visionary responsible for purchasing large numbers of antibodies with the notion that I would find some method or be able to develop some method with which to use thousands of antibodies. And Eileen Dolan, who's the pharmacogenomicist on the project. And I'll also thank the funding sources as well, and our collaborators at MIT for doing modeling, and all the people in my lab. And lastly, for anybody that would like to do micro-Western arrays in their own lab, you can go to the, if you just type in micro-Western arrays on Google or go to our core facility website, you'll find that for $500, you can submit your samples and profile 6 samples against 100 antibodies, and you can find all of the antibodies on our website, or at least several hundred of them that are available, or you can give us your own antibodies. So with that, I'll take any questions, and thank you for your time.
Thank you very much. I think we have time for a couple of questions.
I was just wondering on the effects of the microRNAs, what you found in change in protein. Was there also microRNAs that affected RNAs that affected protein, and what's the proportion?
That's a good question. So this is the most recent of the things we're working on. So they've previously, very recently, published a series of microRNAs associated with drug sensitivity and associated with mRNA expression. And I don't know what the overlap is yet.
There's previous published work saying in mammalian cells, 80% of the changes in proteins are correlated with mRNA changes. That are brought about by microRNAs. But I was wondering, if your data fits that.
So I haven't looked yet. So what our data do suggest so far is that if you compare RNA platforms against themselves, if you were to take the mRNA expression data set measured by DNA microarray versus RNA sequencing, the overlap, the correlation is R of like 0.2 among the experiments, and the correlation between RNA-seq and the protein platform is like 0.4. And so from that perspective, we correlate better with RNA-seq than RNA-seq correlates with expression, but I still don't know if the noise in the technical measurements would allow you to even know what is the correlation between proteins and mRNAs, and what are the correlations between microRNAs, mRNAs, and proteins, given that the technical variation is still pretty large in trying to measure that. In individuals with high microRNA levels, and their mRNA is not affected at any statistically significant level. But again, there's still, I think, the possibility that these are individual artifacts. In many cases, in a large RNA-seq data set, expression array data set, you filter because of a probe mismatch or because you didn't get enough reads or whatever, and you simply focus on what correlated, and somebody only did the experiment once, biologically and technically, and there's probably, if you were to go back and repeat it 8 times, you would find new things in each experiment.
Okay, since I don't see any questions right away and we're running a bit behind, I think